Good day, grade 8 ST students. I hope this video lesson find you in a safe and healthy condition amidst this pandemic. Again, this is Mr. Marin Rodrigo D. Marinos III, your teacher facilitator for the asynchronous online class in biotechnology. Before you continue learning our topic, please be reminded of the following. Number 1. Choose a study space conducive for an online class. Number 2. Set a time for you to finish all the prescribed activities given by your teacher. Number 3. Take notes while watching the video lesson. Number 4. Listen carefully to the instruction given by your teacher. And number 5. Reach out to your teacher with questions relevant to the topic of the video lesson. Since this is an asynchronous online class, your attendance will not be checked on schedule. Your submitted output will serve as your attendance, so make sure to submit all your outputs on time. In addition to those reminders and instructions, you must have the hard copy of the teacher-made activity sheet provided to you a week before the uploading of this video lesson. Browse the activity sheet and check for the missing pages. Read the instructions carefully and reach out to your teacher for some clarifications. So let us now start. Right now, I am showing you a photo montage which I think is very familiar to all of us. On your activity sheet, you're asked to write down 5 thoughts or insights you can share by looking at the picture. You may pause this video at this moment so you can focus on answering this activity. Before proceeding, make sure that you are through with answering this portion of the activity sheet. If yes, you may now continue playing the video lesson. But if not, you can still have your time to finish it. Let us now proceed to the next part of the video lesson. Right now, there are 3 words flashed on your screen as well as on your activity sheet. To test your vocabulary skills, your task is to list down 3 words that are related to each of the words shown on the screen. You may now pause this video at this moment so you can now focus on answering this activity. Before proceeding, make sure that you are through with answering this portion of the activity sheet. If yes, you may now continue playing the video lesson. But if not, you can still have your time to finish it. Let us now start discussing the nature and mechanism involved in the process called polymerase chain reaction. The ability to work with small amounts of DNA is proving valuable in a wide range of scientific endeavors, from paleontology, agriculture, medicine, and even up to criminology. In DNA fingerprinting analysis, as little as 1 microgram of DNA, the amount of DNA in a small drop of blood, can be used to identify and characterize particular sequences of DNA. But there are some instances even that amount of DNA may not be available. In such case, another method called polymerase chain reaction can come to the rescue. With polymerase chain reaction, it is possible to rapidly replicate or amplify selected DNA segments that are initially present in extremely small amount. In only few hours, PCR can make millions or even billions of copies of a particular DNA sequence, thereby producing enough material for DNA fingerprinting and other related applications. Before we proceed to the next part of the discussion, please answer this question. What is the main purpose of the polymerase chain reaction or PCR? Please write your answer on your activity sheet. You may pause this video while answering, and once you're done, you may now proceed to the next part of the video lesson. The complicated multi-protein system that cells use for DNA replication is not required for the PCR method. Neither the origin of replication, nor DNA unwinding proteins, nor the apparatus for lagging strand synthesis are involved. So to facilitate your learning about PCR, let us have a quick review of the enzymes used in DNA replication. On your activity sheet, you're asked to match column A that contains the list of enzymes with column B that lists down the function of each of the enzymes. You may pause this video at this moment so you can focus on answering this activity. Before proceeding, make sure that you are through with answering this portion of the activity sheet. If yes, you may now continue playing the video lesson. But if not, you can still have your time to finish it. One of the key to the simplicity of the PCR is the use of an unusual DNA polymerase called Taq polymerase. This particular polymerase was first isolated from the bacterium Thermus aquaticus, an inhabitant of the hot springs in the Yellowstone National Park in the USA, where water temperature ranges from 70 to 80 degrees Celsius. The optimal temperature for this enzyme is at 72 degrees Celsius, and it is stable at even higher temperature. This property of the Taq polymerase made possible the automation of the polymerase chain reaction. Aside from the Taq polymerase, PCR uses a set of synthetic primers that can set up a chain reaction that produces an exponentially growing population of specific DNA molecules. With all this on hand, Carey Mullis, an American biochemist, was awarded the Nobel Prize for Chemistry in 1993 for his invention of the polymerase chain reaction back in 1986. Before we proceed to the next part of the discussion, please answer this question. What are the two aspects of the PCR that serve as the key to the simplicity of the procedure? Please write your answer on your activity sheet. You may pause this video while answering, and once you're done, you may now proceed to the next part of the video lesson. It is also important to mention that the automation of PCR was also brought by the invention of the thermocycler or thermal cycler, a machine where the multi-step process of heating and cooling of DNA samples can be conducted to facilitate the amplification of the samples. If you open the upper lid of the thermocycler, you can see some slots on the lower heating block just below the upper heating block. Those are the slots where we can place the PCR tubes containing the PCR basic reaction mixture. We can also input the specific temperature range and even the time allotment for each of the cycles of the PCR using the display and function keys. The PCR basic reaction mixture is placed inside a PCR tube. This mixture contains 5 main components: the DNA sample, primers, Taq polymerase, DNTPs or deoxyribonucleotide triphosphate mix, and buffer. Now that we have our PCR basic reaction mixture inside the slots of our thermocycler, we can now discuss the 3-step cycle of the PCR that includes denaturation, annealing, and extension or elongation. In the denaturation process, the temperature in the thermocycler is raised until it reaches a near boiling temperature of 92 to 95 degrees Celsius. This facilitates the denaturation or the separation of the two DNA strands. Once done with the denaturation process, the PCR reaction mixture is then cooled to 45 to 65 degrees Celsius to allow the primers to bind to the complementary regions of the DNA strand being copied. These primers are short single-stranded DNA that contains at least 15 to 20 nucleotides and consists of sequences that are complementary to the sequences located at the two ends of the DNA segments being amplified. Once the primers anneal to the DNA fragments, the temperature is then raised to 72 degrees Celsius and the Taq polymerase goes to work by adding nucleotides to the 3' end of the primer. It takes no more than a few minutes for the Taq polymerase to completely copy the targeted DNA sequences, thereby doubling the amount of DNA. The reaction mixture is heated again to melt the new double helices. More primer is bound to the DNA and the cycle is repeated to double the amount of the DNA. This reaction cycle is repeated as many times as necessary, with each cycle doubling the amount of DNA from the previous cycle. Now that you're familiar with the basic processes involved in PCR, you are now asked to study the graph on your activity sheet. Identify what part of the polymerase chain reaction is being described by the different portions of the graph. Follow the instructions presented in your activity sheet for your answers. You may pause this video at this moment so you can focus on answering this activity. Before proceeding, make sure that you are through with answering this portion of the activity sheet. If yes, you may now continue playing the video lesson. But if not, you can still have your time to finish it. In most cases, 20 to 30 reaction cycles are sufficient to produce the desired quantity of DNA. The theoretical amplification accompanied by n cycle is 2 raised to n, so 6 cycles yields an amplification of 64 amplicons or PCR products, and 20 cycles yields an amplification of a millionfold of almost 1,048,576 amplicons. Before we proceed to the next part of our discussion, please answer this question first. How many amplicons are theoretically produced from a 30 PCR cycle? Please write your answer on your activity sheet. You may pause this video while answering, and once you're done, you may now proceed watching the next part of our video lesson. Now that you're familiar with the nature and mechanism of PCR, it is now the right time to make some connections from the photo montage presented earlier in this video lesson. Right now in this COVID-19 pandemic, diagnosing suspected COVID-19 patient is useful in accounting the possible transmission of the virus. One type of PCR called RT-PCR comes handy in diagnosing this viral disease. RT-PCR commonly used in swab testing is the gold standard for diagnosing COVID-19. This procedure uses an enzyme called reverse transcriptase in combination with polymerase chain reaction and gel electrophoresis. It can be used to compare gene expression between samples and, for this instance, the presence of viral genome from a suspected patient. Since SARS-CoV-2, the causative agent of COVID-19, is an RNA virus, the reverse transcriptase can be handy in producing the complementary DNA or cDNA of the given RNA from the virus. Once synthesized, the sample is now subjected to the regular polymerase chain reaction for sample amplification. After PCR, the amplicons will be subjected to gel electrophoresis that can be used to further analyze the sample. The mechanism behind gel electrophoresis will be further discussed on your next lesson. So right now, PCR is not just a tool for crime solving, but also for medical diagnosis. It serves as one of the workhorses of the medical field to combat this pandemic. To further your understanding about PCR and RT-PCR, you're asked to research about the invention of the Filipino physician-scientist Dr. Uldestura of UP Manila National Institute of Health. Your task is to write a 5-sentence paragraph about the impact of a locally produced PCR kit for our battle against COVID-19. To test your understanding about our lesson, you must answer the assessment on your activity sheet. For Part 1, read and answer each question carefully. Write the capital letter of your answer on the space provided before each number. For question number 1, who developed the polymerase chain reaction? Letter A, Lynn Margulis, B, Carey Mullis, C, Marshall Nirenberg, or D, Dorothy Hodgkin. Question number 2, in the polymerase chain reaction, what machine is used to imitate DNA replication in vitro? Letter A, thermal cycler, B, polymerizer, C, gel electrophoresis, or D, centrifuge. For question number 3, from what bacterium is the heat-stable polymerase used in PCR derived from? Letter A, Thermus aquaticus, B, Bacillus thuringiensis, C, Desulfovibrio, or D, Streptococcus pneumoniae. For question number 4, which of the following processes is not part of the PCR mechanism? A, initiation, B, denaturation, C, annealing, or D, extension or elongation. And for question number 5, in the first process of PCR, what is the temperature range that facilitates the unwinding of the DNA strands? Letter A, 65 to 75 degrees Celsius. Letter B, 92 to 95 degrees Celsius. Letter C, 45 to 65 degrees Celsius. Or letter D, 95 to 112 degrees Celsius. And for the part 2 of your assessment, read each statement carefully. Write A if the statement is correct and B if it is not. Write your answer on the space provided before each number. For statement number 1, during the annealing process, a primer will attach to both strands of the separated DNA. For statement number 2, PCR is a repetitive multi-step process of heating and cooling of DNA samples. For statement number 3, during the extension process of PCR, the DNA polymerase III will synthesize new DNA strands by adding new nucleotides in a 5' to 3' direction. And for statement number 4, after 5 cycles of PCR, you will have 36 newly synthesized DNA strands. And lastly, for statement number 5, glycosidic bonds between base pairs are broken during the denaturation process. And this ends our lesson about polymerase chain reaction. I will now flash some links that you can browse on the internet if you want to further your study about this revolutionary procedure. And before we end the session, please be reminded to finish all the activities from your activity sheet. Once done, make sure to let your parents sign your output before submitting it next week. Again, this is your teacher facilitator in biotechnology, Mr. Marion Rodrigo D. Marinos III, reminding you to stay safe and healthy and see you soon.